Left photo shows a cluster of mushrooms with bell-like domes attached to slender stalks. Middle photo shows a yellowish-orange fungus that grows in a cluster and is lobe-shaped. Right photo is a micrograph that shows a long, slender stalk that branches into long chains of spores that look like a string of beads. Figure 24. Many species of fungus produce the familiar mushroom, A, which is a reproductive structure. This B, coral fungus, displays brightly colored fruiting bodies. This electron micrograph shows C, the spore-bearing structures of Aspergillus, a type of toxic fungus found mostly in soil and plants. Credit mushroom. Modification of work by Chris Wee. Credit coral fungus. Modification of work by Corey Zanker. Credit Aspergillus. Modification of work by Janice Haney-Carr, Robert Simmons, CDC. Scalebar data from Matt Russell. Chapter outline. 24. 1 Characteristics of fungi. 24. 2 Classifications of fungi. 24. 3 Ecology of fungi. 24. 4 Fungal parasites and pathogens. 24. 5 Importance of fungi in human life. The word fungus comes from the Latin word for mushrooms. Indeed, the familiar mushroom is a reproductive structure used by many types of fungi. However, there are also many fungus species that don't produce mushrooms at all. Being eukaryotes, a typical fungal cell contains a true nucleus and many membrane-bound organelles. The kingdom fungi includes an enormous variety of living organisms. While scientists have identified about 135,000 species of fungi, this is only a fraction of the more than 1.5 million species of fungus likely present on Earth. Edible mushrooms, yeasts, black mold, and the producer of the antibiotic penicillin, Penicillium notatum, are all members of the kingdom fungi, which belongs to the domain Eukarya. Learning objectives. By the end of this section, you will be able to do the following. List the characteristics of fungi. Describe the composition of the mycelium. Describe the mode of nutrition of fungi. Explain sexual and asexual reproduction in fungi. Fungi, once considered plant-like organisms, are more closely related to animals than plants. Fungi are not capable of photosynthesis. They are heterotrophic because they use complex organic compounds as sources of energy and carbon. Fungi share a few other traits with animals. Their cell walls are composed of chitin, which is found in the exoskeletons of arthropods. Fungi produce a number of pigments, including melanin, also found in the hair and skin of animals. Like animals, fungi also store carbohydrates as glycogen. However, like bacteria, fungi absorb nutrients across the cell surface and act as decomposers, helping to recycle nutrients by breaking down organic materials to simple molecules. Some fungal organisms multiply only asexually, whereas others undergo both asexual reproduction and sexual reproduction with alternation of generations. Most fungi produce a large number of spores, which are haploid cells that can undergo mitosis to form multicellular, haploid individuals. Fungi often interact with other organisms, forming beneficial or mutualistic associations. For example, most terrestrial plants form symbiotic relationships with fungi. The roots of the plant connect with the underground parts of the fungus, which form mycorrhizae. Through mycorrhizae, the fungus and plant exchange nutrients and water, greatly aiding the survival of both species. Alternatively, lichens are an association between a fungus and its photosynthetic partner, usually an alga. Fungi also cause serious infections in plants and animals. For example, Dutch elm disease, which is caused by the fungus Ophiostoma ulmi, is a particularly devastating type of fungal infestation that destroys many native species of elm, Ulmus sp. By infecting the tree's vascular system. The elm bark beetle acts as a vector, transmitting the disease from tree to tree. Accidentally introduced in the 1900s, the fungus decimated elm trees across the continent. Many European and Asiatic elms are less susceptible to Dutch elm disease than American elms. In humans, fungal infections are generally considered challenging to treat. Unlike bacteria, fungi do not respond to traditional antibiotic therapy since they are eukaryotes. Fungal infections may prove deadly for individuals with compromised immune systems. Fungi have many commercial applications. The food industry uses yeasts in baking, brewing, and cheese and wine making. Many industrial compounds are byproducts of fungal fermentation. Fungi are the source of many commercial enzymes and antibiotics. Although humans have used yeasts and mushrooms since prehistoric times, until recently, the biology of fungi was poorly understood. In fact, up until the mid-20th century, many scientists classified fungi as plants. Fungi, like plants, are mostly sessile and seemingly rooted in place. They possess a stem-like structure similar to plants, as well as having a root-like fungal mycelium in the soil. In addition, their mode of nutrition was poorly understood. Progress in the field of fungal biology was the result of mycology, the scientific study of fungi. Based on fossil evidence, fungi have been found in the Devonian era, about 410 million years ago. However, new findings might place the appearance of the first fungi during the Neoproterozoic era, about 900 million years ago. Molecular biology analysis of the fungal genome demonstrates that fungi are more closely related to animals than plants. Under some current systematic phylogenies, they continue to be a monophyletic group of organisms. Career connection. Mycologist. Mycologists are biologists who study fungi. Historically, mycology was a branch of microbiology, and many mycologists start their careers with a degree in microbiology. To become a mycologist, a bachelor's degree in a biological science, preferably majoring in microbiology, and a master's degree in mycology are minimally necessary. Mycologists can specialize in taxonomy and fungal genomics, molecular and cellular biology, plant pathology, biotechnology, or biochemistry. Some medical microbiologists concentrate on the study of infectious diseases caused by fungi, called mycoses. Mycologists collaborate with zoologists and plant pathologists to identify and control difficult fungal infections, such as the devastating chestnut blight, the mysterious decline in frog populations in many areas of the world, or the deadly epidemic called white nose syndrome, which is decimating bats in the eastern United States. Government agencies hire mycologists as research scientists and technicians to monitor the health of crops, national parks, and national forests. Mycologists are also employed in the private sector by companies that develop chemical and biological control products or new agricultural products, and by companies that provide disease control services. Because of the key role played by fungi in the fermentation of alcohol and the preparation of many important foods, scientists with a good understanding of fungal physiology routinely work in the food technology industry. Oenology, the science of winemaking, relies not only on the knowledge of grape varietals and soil composition, but also on a solid understanding of the characteristics of the wild yeasts that thrive in different winemaking regions. It is possible to purchase yeast strains isolated from specific grape-growing regions. The great French chemist and microbiologist, Louis Pasteur, made many of his essential discoveries working on the humble brewer's yeast, thus discovering the process of fermentation. Cell structure and function. Fungi are eukaryotes and as such have a complex cellular organization. As eukaryotes, fungal cells contain a membrane-bound nucleus. The DNA in the nucleus is represented by multiple linear molecules wrapped around histone proteins, as is observed in other eukaryotic cells. A few types of fungi have accessory genomic structures comparable to bacterial plasmids, loops of DNA. However, the horizontal transfer of genetic information that occurs between one bacterium and another rarely occurs in fungi. Fungal cells also contain mitochondria and a complex system of internal membranes, including the endoplasmic reticulum and Golgi apparatus. Unlike plant cells, fungal cells do not have chloroplasts or chlorophyll. Many fungi display bright colors arising from other cellular pigments, ranging from red to green to black. The poisonous Amanita muscaria, fly agaric, is recognizable by its bright red cap with white patches, figure 24-2. Pigments in fungi are associated with the cell wall and play a protective role against ultraviolet radiation. Some fungal pigments are toxic to humans. Photo shows two large mushrooms, each with a wide white base and a bright red cap. The caps are dotted with small white protrusions. Figure 24, 2 Amanita. The poisonous Amanita muscaria is native to temperate and boreal regions of North America. Credit, Christine Magel. Like plant cells, Fungal cells have a thick cell wall. The rigid layers of fungal cell walls contain complex polysaccharides called chitin and glucans. Chitin, N-acetyl-D-glucosamine, also found in the exoskeleton of arthropods such as insects, gives structural strength to the cell walls of fungi. The wall provides structural support and protects the cell from desiccation and some predators. Fungi have plasma membranes similar to those of other eukaryotes, except that the structure is stabilized by ergosterol, a steroid molecule that replaces the cholesterol found in animal cell membranes. Most members of the kingdom fungi are non-motile. However, flagella are produced by the spores and gametes in the primitive phylum Chytridiomycota. Growth. The vegetative body of a fungus is a unicellular or multicellular thallus. Unicellular fungi are called yeasts. Multicellular fungi produce thread-like hyphae, singular hypha. Dimorphic fungi can change from the unicellular to multicellular state depending on environmental conditions. Saccharomyces cerevisiae, baker's yeast, and Candida species, the agents of thrush, a common fungal infection, are examples of unicellular fungi. Figure 24. 3. Micrograph shows clumps of small blue spheres. Each sphere is about 5 microns across. Figure 24. 3 Candida albicans. Candida albicans is a yeast cell and the agent of candidiasis and thrush. This organism has a similar morphology to coccus bacteria. However, yeast is a eukaryotic organism. Note the nucleus. Credit: modification of work by Dr. Godon Roberstad, CDC. Scalebar data from Matt Russell. Most fungi are multicellular organisms. They display 2 distinct morphological stages, the vegetative and reproductive. The vegetative stage consists of a tangle of hyphae, whereas the reproductive stage can be more conspicuous. The mass of hyphae is a mycelium, figure 24. 4. It can grow on a surface, in soil or decaying material, in a liquid, or even on living tissue. Although individual hyphae must be observed under a microscope, the mycelium of a fungus can be very large, with some species truly being the fungus humungus. The giant Armillaria solidipes, honey mushroom, is considered the largest organism on Earth, spreading across more than 2,000 acres of underground soil in eastern Oregon. It is estimated to be at least 2,400 years old. Photo depicts a light brown fungus growing in a petri dish. The fungus has the appearance of wrinkled round skin surrounded by powdery residue. A hub-like indentation exists at the center of the fungus. Extending from this hub are folds that resemble spokes on a wheel. Figure 24. 4. A fungal mycelium. The mycelium of the fungus Neotestudina rosati can be pathogenic to humans. The fungus enters through a cut or scrape and develops a mycetoma, a chronic subcutaneous infection. Credit: CDC. Most fungal hyphae are divided into separate cells by end walls called septa, singular, septum, figure 24. 5a-c. In most phyla of fungi, tiny holes in the septa allow for the rapid flow of nutrients and small molecules from cell to cell along the hypha. They are described as perforated septa. The hyphae in bread molds, which belong to the phylum Zygomycota, are not separated by septa. Instead, they are formed by large cells containing many nuclei, multinucleate, an arrangement described as coenocytic hyphae, figure 24. 5b. Part A is an illustration of septated hyphae. Cells within the septated hyphae are rectangular. Each cell has its own nucleus and connects to other cells end to end in a long strand. 2 branches occur in the hyphae. Part B is an illustration of coenocytic hyphae. Like the septated hyphae, the coenocytic hyphae consist of long, branched fibers. However, in coenocytic hyphae, there is no separation between the cells or nuclei. Part C is a light micrograph of septated hyphae from Phylophora richardsi. The hyphae consists of a long chain of cells with multiple branches. Each branch is about 3 microns wide and varies from 3 to 20 microns in length. Figure 24. 5 fungal hyphae. Fungal hyphae may be A septated or B coenocytic. Coeno equals common, cytic equals cell, with many nuclei present in a single hypha. A bright field light micrograph of C. Phylophora richardsi shows septa that divide the hyphae. Credit C. Modification of work by Dr. Lucille Jorg, CDC. Scale bar data from Matt Russell. Fungi thrive in environments that are moist and slightly acidic and can grow in dark places or places exposed to light. They vary in their oxygen requirement. Most fungi are obligate aerobes, requiring oxygen to survive. Other species, such as members of the Chytridiomycota that reside in the rumen of cattle, are obligate anaerobes, in that they only use anaerobic respiration because oxygen will disrupt their metabolism or kill them. Yeasts are intermediate, being facultative anaerobes. This means that they grow best in the presence of oxygen using aerobic respiration, but can survive using anaerobic respiration when oxygen is not available. The alcohol produced from yeast fermentation is used in wine and beer production. Nutrition. Like animals, fungi are heterotrophs. They use complex organic compounds as a source of carbon, rather than fix carbon dioxide from the atmosphere, as do some bacteria and most plants. In addition, fungi do not fix nitrogen from the atmosphere. Like animals, they must obtain it from their diet. However, unlike most animals, which ingest food and then digest it internally in specialized organs, fungi perform these steps in the reverse order. Digestion precedes ingestion. First, exoenzymes are transported out of the hyphae, where they process nutrients in the environment. Then, the smaller molecules produced by this external digestion are absorbed through the large surface area of the mycelium. As with animal cells, the polysaccharide of storage is glycogen, a branched polysaccharide rather than amylopectin, a less densely branched polysaccharide, and amylose, a linear polysaccharide, as found in plants. Fungi are mostly saprobes. Saprophyte is an equivalent term. Organisms that derive nutrients from decaying organic matter. They obtain their nutrients from dead or decomposing organic material derived mainly from plants. Fungal exoenzymes are able to break down insoluble compounds, such as the cellulose and lignin of dead wood, into readily absorbable glucose molecules. The carbon, nitrogen, and other elements are thus released into the environment. Because of their varied metabolic pathways, fungi fulfill an important ecological role and are being investigated as potential tools in bioremediation of chemically damaged ecosystems. For example, some species of fungi can be used to break down diesel oil and polycyclic aromatic hydrocarbons, PAHs. Other species take up heavy metals, such as cadmium and lead. Some fungi are parasitic, infecting either plants or animals. Smut and Dutch elm disease affect plants, whereas athlete's foot and candidiasis, thrush, are medically important fungal infections in humans. In environments poor in nitrogen, some fungi resort to predation of nematodes, small non-segmented roundworms. In fact, species of Arthrobatries fungi have a number of mechanisms to trap nematodes. One mechanism involves constricting rings within the network of hyphae. The rings swell when they touch the nematode, gripping it in a tight hold. The fungus then penetrates the tissue of the worm by extending specialized hyphae called haustoria. Many parasitic fungi possess haustoria. As these structures penetrate the tissues of the host, release digestive enzymes within the host's body, and absorb the digested nutrients. Reproduction. Fungi reproduce sexually and/or asexually. Some fungi reproduce both sexually and asexually, while other fungi reproduce only asexually, by mitosis. In both sexual and asexual reproduction, fungi produce spores that disperse from the parent organism by either floating on the wind or hitching a ride on an animal. Fungal spores are smaller and lighter than plant seeds. For example, the giant puffball mushroom bursts open and releases trillions of spores in a massive cloud of what looks like finely particulate dust. The huge number of spores released increases the likelihood of landing in an environment that will support growth. Figure 24. 6. Part A is a photo of a puffball mushroom, which is round and white. Part B is an illustration of a puffball mushroom releasing spores through its exploded top. Figure 24. 6. Puffball and spores. A giant puffball mushroom releases B. a cloud of spores when it reaches maturity. Credit A. Modification of work by Roger Griffith. Credit B. Modification of work by Pearson Scott Forsman, donated to the Wikimedia Foundation. Asexual reproduction. Fungi reproduce asexually by fragmentation, budding, or producing spores. Fragments of hyphae can grow new colonies. Somatic cells in yeast form buds. During budding, an expanded type of cytokinesis, a bulge forms on the side of the cell, the nucleus divides mitotically, and the bud ultimately detaches itself from the mother cell, figure 24. 7. Micrograph shows budding yeast cells. The parent cells are stained dark blue and round, with smaller, teardrop-shaped cells budding from them. The cells are about 2 microns across and 3 microns long. Figure 24. 7. Budding in histoplasma. The dark cells in this bright field light micrograph are the pathogenic yeast Histoplasma capsulatum, seen against a backdrop of light blue tissue. Histoplasma primarily infects lungs but can spread to other tissues, causing histoplasmosis, a potentially fatal disease. Credit: modification of work by Dr. Libero Agelo, CDC. Scalebar data from Matt Russell. The most common mode of asexual reproduction is through the formation of asexual spores, which are produced by a single individual thallus, through mitosis, and are genetically identical to the parent thallus, figure 24. 8. Spores allow fungi to expand their distribution and colonize new environments. They may be released from the parent thallus either outside or within a special reproductive sac called a sporangium. The asexual and sexual stages of reproduction of fungi are shown. In the asexual life cycle, a haploid 1N mycelium undergoes mitosis to form spores. Germination of the spores results in the formation of more mycelia. In the sexual life cycle, the mycelium undergoes plasmagamy, a process in which haploid cells fuse to form a heterokaryon, a cell with 2 or more haploid nuclei. This is called the heterokaryotic stage. The dikaryotic cells, cells with 2 or more nuclei, undergo karyogamy, a process in which the nuclei fuse to form a diploid 2N zygote. The zygote undergoes meiosis to form haploid 1N spores. Germination of the spores results in the formation of a multicellular mycelium. Figure 24. 8. Generalized fungal life cycle. Fungi may have both asexual and sexual stages of reproduction. There are many types of asexual spores. Cnidiospores are unicellular or multicellular spores that are released directly from the tip or side of the hypha. Other asexual spores originate in the fragmentation of a hypha to form single cells that are released as spores. Some of these have a thick wall surrounding the fragment, yet others bud off the vegetative parent cell. In contrast to cnidiospores, sporangiospores are produced directly from a sporangium, figure 24. 9. Micrograph shows several long, thread-like hyphae stained blue. One hypha has a round sporangium, about 35 microns in diameter, at the tip. The sporangium is dark blue at the neck and grainy white and blue elsewhere. Spores that have already been released appear as small white ovals. Figure 24. 9 sporangiospores. This bright field light micrograph shows the release of spores from a sporangium at the end of a hypha called a sporangiophore. The organism is a mucor sp. Fungus, a mold often found indoors. Credit: modification of work by Dr. Lucille Jorg, CDC, scalebar data from Matt Russell. Sexual reproduction. Sexual reproduction introduces genetic variation into a population of fungi. In fungi, sexual reproduction often occurs in response to adverse environmental conditions. During sexual reproduction, 2 mating types are produced. When both mating types are present in the same mycelium, it is called homothallic or self-fertile. Heterothallic mycelia require 2 different but compatible mycelia to reproduce sexually. Although there are many variations in fungal sexual reproduction, all include the following 3 stages, figure 24. 8. First, during plasmagamy, literally, marriage or union of cytoplasm. 2 haploid cells fuse, leading to a dikaryotic stage where 2 haploid nuclei coexist in a single cell. During karyogamy, nuclear marriage, the haploid nuclei fuse to form a diploid zygote nucleus. Finally, meiosis takes place in the gametangia, singular gametangium organs, in which gametes of different mating types are generated. At this stage, spores are disseminated into the environment. Link to learning. Review the characteristics of fungi by watching this video. Learning objectives. By the end of this section, you will be able to do the following. Identify fungi and place them into the 5 major phyla according to current classification. Describe each phylum in terms of major representative species and patterns of reproduction. The kingdom Fungi contains 5 major phyla that were established according to their mode of sexual reproduction or using molecular data. Unrelated fungi that reproduce without a sexual cycle were once placed for convenience in a 6th group, the Deuteramycota, called a form phylum. Because superficially they appeared to be similar, a polyphyletic group of organisms that is of mixed evolutionary origin. However, most mycologists have discontinued this practice. Rapid advances in molecular biology and the sequencing of 18S rRNA, ribosomal RNA, continue to show new and different relationships among the various categories of fungi. The 5 true phyla of fungi are the Chytridiomycota, chytrids, the Zygomycota, conjugated fungi, the Escamycota, sac fungi, the Basidiomycota, club fungi, and the recently described phylum Glomeramycota. Figure 24. 10. The image displays the phylogenetic tree for fungi. From a central source, branches include Basidiomycota and Escamycota, which exist at the end of the same branch, Glomeramycota, Zygomycota, and Chytridiomycota. Animalia are diploid coming from a separate branch from the same source ancestor, but are not a part of the fungi. Figure 24. 10 fungal phyla. Note: Mycota is used to designate a phylum, while mycetes formally denotes a class or is used informally to refer to all members of the phylum. Chytridiomycota, the chytrids, the only class in the phylum Chytridiomycota is the Chytridiomycetes. The chytrids are the simplest and most primitive Eumycota, or true fungi. The evolutionary record shows that the first recognizable chytrids appeared during the late Precambrian period, more than 500 million years ago. Like all fungi, chytrids have chitin in their cell walls, but one group of chytrids has both cellulose and chitin in the cell wall. Most chytrids are unicellular. However, a few form multicellular organisms and hyphae, which have no septa between cells, coenocytic. The chytrids are the only fungi that have retained flagella. They produce both gametes and diploid zoospores that swim with the help of a single flagellum. An unusual feature of the chytrids is that both male and female gametes are flagellated. The ecological habitat and cell structure of chytrids have much in common with protists. Chytrids usually live in aquatic environments, although some species live on land. Some species thrive as parasites on plants, insects, or amphibians, figure 24. 11, while others are saprobes. The chytrid species Alamyces is well characterized as an experimental organism. Its reproductive cycle includes both asexual and sexual phases. Alamyces produces diploid or haploid flagellated zoospores in a sporangium. Micrograph A shows an arthropod, which is a teardrop-shaped transparent organism about 90 microns across and 120 microns long. Tentacle-like appendages jut out from the front, wide end of the organism, and clusters of cilia-like appendages jut out from either side and the back. Transparent oval organisms about 20 microns across cling to the arthropod. Micrograph B shows a similar oval transparent organisms clinging to rod-shaped algae about 5 microns across and 200 microns long. Figure 24. 11 chytrids. The chytrid Batrachochytrium dendrobatides is seen in these light micrographs as transparent spheres growing on A, a freshwater arthropod, water mite, and B, algae. This chytrid causes skin diseases in many species of amphibians, resulting in species decline and extinction. Credit: modification of work by Johnson M.L., Spear R., CDC. Zygomycota, the conjugated fungi. The zygomycetes are a relatively small group of fungi belonging to the phylum Zygomycota. They include the familiar bread mold, Rhizopus stolonifer, which rapidly propagates on the surfaces of breads, fruits, and vegetables. Most species are saprobes, living off decaying organic material. A few are parasites, particularly of insects. Zygomycetes play a considerable commercial role. For example, the metabolic products of some species of Rhizopus are intermediates in the synthesis of semisynthetic steroid hormones. Zygomycetes have a thallus of coenocytic hyphae in which the nuclei are haploid when the organism is in the vegetative stage. The fungi usually reproduce asexually by producing sporangiospores, figure 24. 12. The black tips of bread mold are the swollen sporangia packed with black spores, figure 24. 13. When spores land on a suitable substrate, they germinate and produce a new mycelium. Sexual reproduction starts when environmental conditions become unfavorable. Two opposing mating strains, type plus and type, must be in close proximity for gametangia from the hyphae to be produced and fuse, leading to karyogamy. Each zygospore can contain several diploid nuclei. The developing diploid zygospores have thick coats that protect them from desiccation and other hazards. They may remain dormant until environmental conditions are favorable. When the zygospore germinates, it undergoes meiosis and produces haploid spores, which will, in turn, grow into a new organism. This form of sexual reproduction in fungi is called conjugation, although it differs markedly from conjugation in bacteria and protists, giving rise to the name conjugated fungi. The asexual and sexual life cycles of zygomycetes are shown. In the asexual life cycle, 1N spores undergo mitosis to form long chains of cells called mycelia. Germination results in the formation of more spores. In the sexual life cycle, spores germinate to form mycelia with two different mating types, plus and minus. If the plus and minus mating types are in close proximity, extensions called gametangia form between them. In a process called plasmagamy, the gametangia fuse to form a zygosporangium with multiple haploid nuclei. A thick, protective coat forms around the zygosporangium. In a process called karyogamy, the nuclei fuse to form a zygote with multiple diploid 2N nuclei. The zygote undergoes meiosis and germination. A sporangium grows on a short stalk. Haploid spores are formed inside. The spores germinate, ending the cycle. Figure 24. 12 zygomycete life cycle. Zygomycetes have asexual and sexual phases in their life cycles. In the asexual phase, spores are produced from haploid sporangia by mitosis, not shown. In the sexual phase, plus and minus haploid mating types conjugate to form a heterokaryotic zygosporangium. Karyogamy then produces a diploid zygote. Diploid cells in the zygote undergo meiosis and germinate to form a haploid sporangium, which releases the next generation of haploid spores. The photo in Part A shows a thick layer of green mold growing on bread. Fuzzy white projections grow from the mold. Part B shows a photo of black tips in bread mold. Figure 24. 13 rhizopus spores. Asexual sporangia grow at the end of stalks, which appear as a white fuzz seen on this bread mold, Rhizopus stolonifer. The black tips B of bread mold are the spore-containing sporangia. Credit B, modification of work by Pollandez/Flicker. Ischamycota, the sac fungi. The majority of known fungi belong to the phylum Ischamycota, which is characterized by the formation of an ascus, plural, asci, a sac-like structure that contains haploid ascospores. Filamentous ascomycetes produce hyphae divided by perforated septa, allowing streaming of cytoplasm from one cell to another. Conidia and asci, which are used respectively for asexual and sexual reproduction, are usually separated from the vegetative hyphae by blocked, non-perforated septa. Many ascomycetes are of commercial importance. Some play a beneficial role for humanity, such as the yeasts used in baking, brewing, and wine fermentation, and directly as food delicacies such as truffles and morels. Aspergillus oryzae is used in the fermentation of rice to produce sake. Other ascomycetes parasitize plants and animals, including humans. For example, fungal pneumonia poses a significant threat to AIDS patients who have a compromised immune system. Ascomycetes not only infest and destroy crops directly, they also produce poisonous secondary metabolites that make crops unfit for consumption. Asexual reproduction is frequent and involves the production of conidiophores that release haploid conidiospores. Figure 24. 14. Sexual reproduction starts with the development of special hyphae from either one of two types of mating strains. Figure 24. 14. The male strain produces an antheridium and the female strain develops an escagonium. At fertilization, the antheridium and the escagonium combine in plasmagamy without nuclear fusion. Special dikaryotic escogenous, ascus-producing, hyphae arise from this dikarion, in which each cell has pairs of nuclei. one from the male strain and one from the female strain. In each ascus, 2 haploid nuclei fuse in karyogamy. Thousands of asci fill a fruiting body called the ascocarp. The diploid nucleus in each ascus gives rise to haploid nuclei by meiosis, and spore walls form around each nucleus. The spores in each ascus contain the meiotic products of a single diploid nucleus. The ascospores are then released, germinate, and form hyphae that are disseminated in the environment and start new mycelia, figure 24. 15. Visual connection. Ascomycetes have both sexual and asexual life cycles. In the asexual life cycle, the haploid 1N mycelium branches into a chain of cells called the kinidiophore. Spores bud from the end of the kinidiophore and germinate to form more mycelia. In the sexual life cycle, a round structure called an antheridium buds from the male strain, and a similar structure called the ascogonium buds from the female strain. In a process called plasmagamy, the ascogonium and antheridium fuse to form a cell with multiple haploid nuclei. Mitosis and cell division result in the growth of many hyphae, which form a fruiting body called the ascocarp. The hyphae are dikaryotic, meaning they have 2 haploid nuclei. Asci form at the tips of these hyphae. In a process called karyogamy, the nuclei in the asci fuse to form a diploid 2N zygote. The zygote undergoes meiosis without cell division, resulting in an ascus with 4 1N nuclei arranged in a row. Each nucleus undergoes mitosis, resulting in 8 ascospores, which are also arranged in a row at the tip of the hyphae. Dispersal and germination results in the growth of new mycelia. Figure 24. 14. Ascomycete life cycle. The life cycle of an ascomycete is characterized by the production of asci during the sexual phase. In each ascus, the 4 nuclei produced by meiosis divide once mitotically for a total of 8 haploid ascospores. The haploid phase is the predominant phase of the life cycle in ascomycetes. Which of the following statements is true? A dikaryotic ascus that forms in the ascocarp undergoes karyogamy, meiosis, and mitosis to form 8 ascospores. A diploid ascus that forms in the ascocarp undergoes karyogamy, meiosis, and mitosis to form 8 ascospores. A haploid zygote that forms in the ascocarp undergoes karyogamy, meiosis, and mitosis to form 8 ascospores. A dikaryotic ascus that forms in the ascocarp undergoes plasmagamy, meiosis, and mitosis to form 8 ascospores. Micrograph shows asci, which appear as multiple, sphere-like shapes fused together into a structure about 7 microns across, and ascospores, which are small, light blue ovals about 2 microns wide by 3 microns long released from the asci. Figure 24. 15 ascospores. The bright field light micrograph shows ascospores being released from asci in the fungus Talaromyces flavus var. Flavus. Credit, modification of work by Dr. Lucille Jorg, CDC. Scalebar data from Matt Russell. Basidiomycota, the club fungi. The fungi in the phylum Basidiomycota are easily recognizable under a light microscope by their club-shaped fruiting bodies called basidia, singular, basidium, which are the swollen terminal cells of hyphae. The basidia, which are the reproductive organs of these fungi, are often contained within the familiar mushroom, commonly seen in fields after rain, on the supermarket shelves, and growing on your lawn, figure 24. 16. These mushroom-producing basidiomycetes are sometimes referred to as gill fungi because of the presence of gill-like structures on the underside of the cap. The gills are actually compacted hyphae on which the basidia are born. This group also includes shelf fungi, which cling to the bark of trees like small shelves. In addition, the Basidiomycota includes smuts and rusts, which are important plant pathogens. Most edible fungi belong to the phylum Basidiomycota. However, some Basidiomycota are inedible and produce deadly toxins. For example, Cryptococcus neoformans causes severe respiratory illness. The infamous death cap mushroom, Amanita phalloides, is related to the fly agaric seen at the beginning of the previous section. Photo shows toadstool mushrooms growing on a lawn and forming a large circle. Figure 24. 16 fairy ring. The fruiting bodies of a basidiomycete form a ring in a meadow, commonly called fairy ring. The best-known fairy ring fungus has the scientific name Marasmius oreads. The body of this fungus, its mycelium, is underground and grows outward in a circle. As it grows, the mycelium depletes the soil of nitrogen, causing the mycelia to grow away from the center and leading to the fairy ring of fruiting bodies where there is adequate soil nitrogen. Credit, CropCircles/Wikipedia Commons. The life cycle of basidiomycetes includes sexual and asexual reproduction, figure 24:17. Most fungi are haploid through most of their life cycles, but the basidiomycetes produce both haploid and dikaryotic mycelia, with the dikaryotic phase being dominant. Note, the dikaryotic phase is technically not diploid, since the nuclei remain unfused until shortly before spore production. In the basidiomycetes, sexual spores are more common than asexual spores. The sexual spores form in the club-shaped basidium and are called basidiospores. In the basidium, nuclei of two different mating strains fuse, karyogamy, giving rise to a diploid zygote that then undergoes meiosis. The haploid nuclei migrate into 4 different chambers appended to the basidium and then become basidiospores. Each basidiospore germinates and generates monokaryotic haploid hyphae. The mycelium that results is called a primary mycelium. Mycelia of different mating strains can combine and produce a secondary mycelium that contains haploid nuclei of 2 different mating strains. This is the dominant dikaryotic stage of the basidiomycete life cycle. Thus, each cell in this mycelium has 2 haploid nuclei, which will not fuse until formation of the basidium. Eventually, the secondary mycelium generates a basidiocarp, a fruiting body that protrudes from the ground. This is what we think of as a mushroom. The basidiocarp bears the developing basidia on the gills under its cap. Visual connection: the life cycle of basidiomycetes, better known as mushrooms, is shown. Basidiomycetes have a sexual life cycle that begins with the germination of 1N basidiospores into mycelia with plus and minus mating types. In a process called plasmagamy, the plus and minus mycelia form a dikaryotic mycelium. Under the right conditions, the dikaryotic mycelium grows into a bastiocarp, or mushroom. Gills on the underside of the mushroom cap contain cells called basidia. The basidia undergo karyogamy to form a 2N zygote. The zygote undergoes meiosis to form cells with 4 haploid, 1N, nuclei. Cell division results in 4 basidiospores. Dispersal and germination of basidiospores ends the cycle. Figure 24:17 Basidiomycete life cycle. The life cycle of a basidiomycete has sexual and asexual reproduction with haploid and dikaryotic mycelia. Haploid primary mycelia fuse to form a dikaryotic secondary mycelium, which is the dominant stage of the life cycle and produces the basidiocarp. Which of the following statements is true? A basidium is the fruiting body of a mushroom-producing fungus, and it forms 4 basidiocarps. The result of the plasmagamy step is 4 basidiospores. Karyogamy results directly in the formation of mycelia. A basidiocarp is the fruiting body of a mushroom-producing fungus. Asexual Escamacota and Basidiomycota. Imperfect fungi, those that do not display a sexual phase, were formerly classified in the form phylum Deuteromycota, an invalid taxon no longer used in the present, ever-developing classification of organisms. While Deuteromycota was once a classification taxon, recent molecular analysis has shown that some of the members classified in this group belong to the Escamacota, figure 24:18, or the Basidiomycota. Because some members of this group have not yet been appropriately classified, they are less well described in comparison to members of other fungal taxa. Most imperfect fungi live on land, with a few aquatic exceptions. They form visible mycelia with a fuzzy appearance and are commonly known as mold. Micrograph shows aspergillus mycelia, which look like long threads, and a spherical conidiophore about 40 microns across. Figure 24:18 Aspergillus. Aspergillus niger is an asexually reproducing fungus, phylum Escamacota, commonly found as a food contaminant. The spherical structure in this light micrograph is an asexual conidiophore. Molecular studies have placed Aspergillus with the ascomycetes, and sexual cycles have been identified in some species. Credit: modification of work by Dr. Lucille Jorg, CDC. Scalebar data from Matt Russell. The fungi in this group have a large impact on everyday human life. The food industry relies on them for ripening some cheeses. The blue veins in Roquefort cheese and the white crust on camembert are the result of fungal growth. The antibiotic penicillin was originally discovered on an overgrown Petri plate, on which a colony of Penicillium fungi had killed the bacterial growth surrounding it. Other fungi in this group cause serious diseases, either directly as parasites, which infect both plants and humans, or as producers of potent toxic compounds, as seen in the aflatoxins released by fungi of the genus Aspergillus. Glomeramycota. The Glomeramycota is a newly established phylum that comprises about 230 species, all of which are involved in close associations with the roots of trees. Fossil records indicate that trees and their root symbionts share a long evolutionary history. It appears that nearly all members of this family form arbuscular mycorrhizae. The hyphae interact with the root cells, forming a mutually beneficial association in which the plants supply the carbon source and energy in the form of carbohydrates to the fungus, and the fungus supplies essential minerals from the soil to the plant. The exception is Geosiphon piriformis, which hosts the Cyanobacterium nostoc as an endosymbiont. The glomeromycetes do not reproduce sexually and do not survive without the presence of plant roots. Although they have coenocytic hyphae like the zygomycetes, they do not form zygospores. DNA analysis shows that all glomeromycetes probably descended from a common ancestor, making them a monophyletic lineage. Learning objectives. By the end of this section, you will be able to do the following. Describe the role of fungi in various ecosystems. Describe mutualistic relationships of fungi with plant roots and photosynthetic organisms. Describe the beneficial relationship between some fungi and insects. Fungi play a crucial role in the constantly changing balance of ecosystems. They colonize most habitats on Earth, preferring dark, moist conditions. They can thrive in seemingly hostile environments, such as the tundra, thanks to a most successful symbiosis with photosynthetic organisms like algae to produce lichens. Within their communities, fungi are not as obvious as are large animals or tall trees. Like bacteria, they act behind the scenes as major decomposers. With their versatile metabolism, fungi break down organic matter, which would otherwise not be recycled. Habitats. Although fungi are primarily associated with humid and cool environments that provide a supply of organic matter, they colonize a surprising diversity of habitats, from seawater to human skin and mucous membranes. Chytrids are found primarily in aquatic environments. Other fungi, such as Coccidioides immitis, which causes pneumonia when its spores are inhaled, thrive in the dry and sandy soil of the southwestern United States. Fungi that parasitize coral reefs live in the ocean. However, most members of the kingdom fungi grow on the forest floor, where the dark and damp environment is rich in decaying debris from plants and animals. In these environments, fungi play a major role as decomposers and recyclers, making it possible for members of the other kingdoms to be supplied with nutrients and live. Decomposers and recyclers. The food web would be incomplete without organisms that decompose organic matter. Figure 24. 19. Some elements, such as nitrogen and phosphorus, are required in large quantities by biological systems and yet are not abundant in the environment. The action of fungi releases these elements from decaying matter, making them available to other living organisms. Trace elements present in low amounts in many habitats are essential for growth and would remain tied up in rotting organic matter if fungi and bacteria did not return them to the environment via their metabolic activity. Photo shows two shell-shaped mushrooms growing on decaying wood. Figure 24. 19 bracket fungi. Fungi are an important part of ecosystem nutrient cycles. These bracket fungi growing on the side of a tree are the fruiting structures of a basidiomycete. They receive their nutrients through their hyphae, which invade and decay the tree trunk. Credit. Cory Zanker. The ability of fungi to degrade many large and insoluble molecules is due to their mode of nutrition. As seen earlier, digestion precedes ingestion. Fungi produce a variety of exoenzymes to digest nutrients. The enzymes are either released into the substrate or remain bound to the outside of the fungal cell wall. Large molecules are broken down into small molecules, which are transported into the cell by a system of protein carriers embedded in the cell membrane. Because the movement of small molecules and enzymes is dependent on the presence of water, active growth depends on a relatively high percentage of moisture in the environment. As saprobes, fungi help maintain a sustainable ecosystem for the animals and plants that share the same habitat. In addition to replenishing the environment with nutrients, fungi interact directly with other organisms in beneficial, and sometimes damaging, ways. Figure 24. 20. Photo shows a shell-shaped shelf fungus growing up the trunk of a living tree. Figure 24. 20. Shelf fungi. Shelf fungi, so called because they grow on trees in a stack, attack and digest the trunk or branches of a tree. While some shelf fungi are found only on dead trees, others can parasitize living trees and cause eventual death, so they are considered serious tree pathogens. Credit: Corey Zanker. Mutualistic relationships. Symbiosis is the ecological interaction between two organisms that live together. This definition does not describe the type or quality of the interaction. When both members of the association benefit, the symbiotic relationship is called mutualistic. Fungi form mutualistic associations with many types of organisms, including cyanobacteria, algae, plants, and animals. Fungus-plant mutualism. One of the most remarkable associations between fungi and plants is the establishment of mycorrhizae. Mycorrhiza, which is derived from the Greek words myco meaning fungus and rhizo meaning root, refers to the fungal partner of a mutualistic association between vascular plant roots and their symbiotic fungi. Nearly 90% of all vascular plant species have mycorrhizal partners. In a mycorrhizal association, the fungal mycelia use their extensive network of hyphae and large surface area in contact with the soil to channel water and minerals from the soil into the plant. In exchange, the plant supplies the products of photosynthesis to fuel the metabolism of the fungus. There are several basic types of mycorrhizae. Ectomycorrhizae, outside mycorrhizae, depend on fungi enveloping the roots in a sheath called a mantle. Hyphae grow from the mantle into the root and envelope the outer layers of the root cells in a network of hyphae called a Hardig net. Figure 24. 21. The fungal partner can belong to the Escamycota, Basidiomycota, or Zygomycota. Endomycorrhizae, inside mycorrhizae, also called arbuscular mycorrhizae, are produced when the fungi grow inside the root in a branched structure called an arbuscule, from the Latin for little trees. The fungal partners of endomycorrhizal associates all belong to the Glomeromycota. The fungal arbuscules penetrate root cells between the cell wall and the plasma membrane, and are the site of the metabolic exchanges between the fungus and the host plant. Figure 24. 21b and Figure 24. 22b. Orchids rely on a third type of mycorrhiza. Orchids are epiphytes that typically produce very small airborne seeds without much storage to sustain germination and growth. Their seeds will not germinate without a mycorrhizal partner, usually a basidiomycete. After nutrients in the seed are depleted, fungal symbionts support the growth of the orchid by providing necessary carbohydrates and minerals. Some orchids continue to be mycorrhizal throughout their life cycle. Visual connection. The figure compares two types of mycorrhizae, ectomycorrhiza and arbuscular mycorrhiza. Part A is an illustration of an ectomycorrhiza mycorrhiza. In ectomycorrhiza, fungal hyphae form a structure called a Hardig net inside the root. The Hardig net forms rows of cells that extend straight down and branch toward the outside of the root. A fungal mantle surrounds the root. Mycelia extend from the fungal mantle. In arbuscular mycorrhiza, the fungi form finger-like clusters that are connected to mycelia that extend from the root into the soil. Part B is a micrograph of arbuscular mycorrhiza, which appear as grape-like clusters in a root tip. Figure 24. 21. Two types of mycorrhizae. A. Ectomycorrhizae and B. Arbuscular or endomycorrhizae have different mechanisms for interacting with the roots of plants. Credit B. M.S. Termal, University of Manitoba, Plant Science Department. If symbiotic fungi were absent from the soil, what impact do you think this would have on plant growth? Part A is a photograph showing a white fungal mantle that covers a tree-like structure that has grown from the side of a root. Part B is a micrograph showing ribbon-like hyphae surrounded by spores that are about 30 microns across. Figure 24. 22 mycorrhizae. The infection of Pinus radiata, Monterey pine, roots by the hyphae of Amanita muscaria, fly Amanita, causes the pine tree to produce many small, branched rootlets. The Amanita hyphae cover these small roots with a white mantle. B. Spores, the round bodies and hyphae, thread-like structures, are evident in this light micrograph of an arbuscular mycorrhiza by a fungus on the root of a corn plant. Credit A. Modification of work by Randy Molina, USDA. Credit B. Modification of work by Sarah Wright, USDA GRS. Scale bar data from Matt Russell. Other examples of fungus, plant mutualism, include the endophytes, fungi that live inside tissue without damaging the host plant. Endophytes release toxins that repel herbivores or confer resistance to environmental stress factors, such as infection by microorganisms, drought, or heavy metals in soil. Evolution connection, coevolution of land plants, and mycorrhizae. As we have seen, mycorrhizae are the fungal partners of a mutually beneficial symbiotic association that co-evolved between roots of vascular plants and fungi. A well-supported theory proposes that fungi were instrumental in the evolution of the root system in plants and contributed to the success of angiosperms. The bryophytes, mosses, and liverworts, which are considered the most ancestral plants and the first to survive and adapt on land, have simple underground rhizoids rather than a true root system, and therefore cannot survive in dry areas. However, some bryophytes have arbuscular mycorrhizae, and some do not. True roots first appeared in the ancestral vascular plants. Vascular plants that developed a system of thin extensions from their roots would have had a selective advantage over non-vascular plants because they had a greater surface area of contact with the fungal partners than did the rhizoids of mosses and liverworts. The first true roots would have allowed vascular plants to obtain more water and nutrients in the ground. Fossil records indicate that fungi actually preceded the invasion of ancestral freshwater plants onto dry land. The first association between fungi and photosynthetic organisms on land involved moss-like plants and endophytes. These early associations developed before roots appeared in plants. Slowly, the benefits of the endophyte and rhizoid interactions for both partners led to present-day mycorrhizae. About 90% of today's vascular plants have associations with fungi in their rhizosphere. The fungi involved in mycorrhizae display many characteristics of ancestral fungi. They produce simple spores, show little diversification, do not have a sexual reproductive cycle, and cannot live outside of a mycorrhizal association. The plants benefited from the association because mycorrhizae allowed them to move into new habitats and allowed the increased uptake of nutrients, which gave them an enormous selective advantage over plants that did not establish symbiotic relationships. Lichens. Lichens display a range of colors and textures, figure 24, 23, and can survive in the most unusual and hostile habitats. They cover rocks, gravestones, tree bark, and the ground in the tundra where plant roots cannot penetrate. Lichens can survive extended periods of drought when they become completely desiccated and then rapidly become active once water is available again. Link to learning. Explore the world of lichens using this site from Oregon State University. Different lichens are shown. Part A shows a lichen that appears like brown flecks on gray rock. Part B shows a moss-like lichen hanging from a tree. Part C shows lichen that have a wide, flat, convoluted shape. Figure 24. 23 lichens. Lichens have many forms. They may be a crust-like, B hair-like, or C leaf-like. Credit A. Modification of work by Joe Naylor. Credit B. Modification of work by DJP Mapleferaman/Flicker. Credit C. Modification of work by Corey Zanker. It is important to note that lichens are not a single organism, but rather another wonderful example of a mutualism in which a fungus, usually a member of the Escamicota or Basidiomycota, lives in a physical and physiological relationship with a photosynthetic organism, a eukaryotic alga or a prokaryotic cyanobacterium. Figure 24. 24. Generally, neither the fungus nor the photosynthetic organism can survive alone outside of the symbiotic relationship. The body of a lichen, referred to as a thallus, is formed of hyphae wrapped around the photosynthetic partner. The photosynthetic organism provides carbon and energy in the form of carbohydrates. Some cyanobacteria additionally fix nitrogen from the atmosphere, contributing nitrogenous compounds to the association. In return, the fungus supplies minerals and protection from dryness and excessive light by encasing the algae in its mycelium. The fungus also attaches the lichen to its substrate. The lichen has multiple layers. The top layer, or upper cortex, is made up of compact fungal hyphae that provide protection to the lichen. Beneath this layer is the algal zone, where fungal hyphae wrap around the photosynthetic algal cells. Below the algal zone is a layer known as the medulla, which is comprised of more loosely arranged fungal hyphae. Beneath the medulla lies the lower cortex, which, like the upper cortex, is comprised of compact fungal hyphae that provide protection to the lichen. Projections beneath the lower cortex anchor the lichen to its substrate. Figure 24. 24 structure of a lichen. This cross-section of a lichen thallus shows the A upper cortex of fungal hyphae, which provides protection, the B algal zone where photosynthesis occurs, the C medulla of fungal hyphae, and the D lower cortex, which also provides protection and may have E rhizines to anchor the thallus to the substrate. The thallus of lichens grows very slowly, expanding its diameter a few millimeters per year. Both the fungus and the alga participate in the formation of dispersal units, called sordia, clusters of algal cells surrounded by mycelia. Sordia are dispersed by wind and water and form new lichens. Lichens are extremely sensitive to air pollution, especially to abnormal levels of nitrogenous and sulfurous compounds. The U.S. Forest Service and National Park Service can monitor air quality by measuring the relative abundance and health of the lichen population in an area. Lichens fulfill many ecological roles. Caribou and reindeer eat lichens, and they provide cover for small invertebrates that hide in the mycelium. In the production of textiles, weavers used lichens to dye wool for many centuries until the advent of synthetic dyes. The pigments used in litmus paper are also extracted from lichens. Link to learning. Lichens are used to monitor the quality of air. Read more on this site from the United States Forest Service. Fungus-animal mutualism. Fungi have evolved mutualisms with numerous insects in phylum Arthropoda, joint-legged invertebrates with a chitinous exoskeleton. Arthropods depend on the fungus for protection from predators and pathogens, while the fungus obtains nutrients and a way to disseminate spores into new environments. The association between species of Basidiomycota and scale insects is one example. The fungal mycelium covers and protects the insect colonies. The scale insects foster a flow of nutrients from the parasitized plant to the fungus. In a second example, leafcutter ants of Central and South America literally farm fungi. They cut discs of leaves from plants and pile them up in subterranean gardens, figure 24. 25. Fungi are cultivated in these disc gardens, digesting the cellulose in the leaves that the ants cannot break down. Once smaller sugar molecules are produced and consumed by the fungi in turn become a meal for the ants. The insects also patrol their garden, preying on competing fungi. Both ants and fungi benefit from this mutualistic association. The fungus receives a steady supply of leaves and freedom from competition, while the ants feed on the fungi they cultivate. Photo shows an ant carrying a leaf over its head. Figure 24. 25 leafcutter ant. A leafcutter ant transports a leaf that will feed a farmed fungus. Credit: Scott Bauer, USDA-RS. Fungivores. Animal dispersal is important for some fungi, because an animal may carry fungal spores considerable distances from the source. Fungal spores are rarely completely degraded in the gastrointestinal tract of an animal, and many are able to germinate when they are passed in the feces. Some dung fungi actually require passage through the digestive system of herbivores to complete their life cycle. The black truffle, a prized gourmet delicacy, is the fruiting body of an underground ascomycete. Almost all truffles are ectomycorrhizal and are usually found in close association with trees. Animals eat truffles and disperse the spores. In Italy and France, truffle hunters use female pigs to sniff out truffles. Female pigs are attracted to truffles because the fungus releases a volatile compound closely related to a pheromone produced by male pigs. Learning objectives. By the end of this section, you will be able to do the following. Describe some fungal parasites and pathogens of plants. Describe the different types of fungal infections in humans. Explain why antifungal therapy is hampered by the similarity between fungal and animal cells. Parasitism describes a symbiotic relationship in which one member of the association benefits at the expense of the other. Both parasites and pathogens harm the host. However, pathogens cause disease, damage to host tissues or physiology, whereas parasites usually do not, but can cause serious damage and death by competition for nutrients or other resources. Commensalism occurs when one member benefits without affecting the other. Plant parasites and pathogens. The production of sufficient high-quality crops is essential to human existence. Unfortunately, plant diseases have ruined many crops throughout human agricultural history, sometimes creating widespread famine. Many plant pathogens are fungi that cause tissue decay and the eventual death of the host. Figure 24. 26. In addition to destroying plant tissue directly, some plant pathogens spoil crops by producing potent toxins that can further damage and kill the host plant. Fungi are also responsible for food spoilage and the rotting of stored crops. For example, the fungus Claviceps purpurea causes ergot, a disease of cereal crops, especially of rye. Although the fungus reduces the yield of cereals, the effects of the ergot's alkaloid toxins on humans and animals are of much greater significance. In animals, the disease is referred to as ergotism. The most common signs and symptoms are convulsions, hallucination, gangrene, and loss of milk in cattle. The active ingredient of ergot is lysergic acid, which is a precursor of the drug LSD. Smuts, rusts, and powdery or downy mildew are other examples of common fungal pathogens that affect crops. Image displays various plants and fruits infected with fungal pathogens. A. Green mold on grapefruit. B. Powdery mildew covering zinnia flower and leaves. C. Red-colored rust on barley. D. Gray rot on grapes, which appears as a fibrous, almost cotton-like substance. Figure 24. 26 fungal pathogens. Some fungal pathogens include a green mold on grapefruit, B. Powdery mildew on a zinnia, C. Stem rust on a sheaf of barley, and D. Gray rot on grapes. In wet conditions, Botrytis cinerea, the fungus that causes gray rot, can destroy a grape crop. However, controlled infection of grapes by botrytis results in noble rot, a condition that produces strong and much-prized dessert wines. Credit A. Modification of work by Scott Bauer, USDA-RS. Credit B. Modification of work by Stephen Osmus, USDA-RS. Credit C. Modification of work by David Marshall, USDA-RS. Credit D. Modification of work by Joseph Smilanic, USDA-RS. Aflatoxins are toxic, carcinogenic compounds released by fungi of the genus Aspergillus. Periodically, harvests of nuts and grains are tainted by aflatoxins, leading to massive recall of produce. This sometimes ruins producers and causes food shortages in developing countries. Animal and human parasites and pathogens. Fungi can affect animals, including humans, in several ways. A mycosis is a fungal disease that results from infection and direct damage due to the growth and infiltration of the fungus. Fungi attack animals directly by colonizing and destroying tissues. Mycotoxicosis is the poisoning of humans and other animals by foods contaminated by fungal toxins, mycotoxins. Mycetismus specifically describes the ingestion of preformed toxins in poisonous mushrooms. In addition, individuals who display hypersensitivity to molds and spores may develop strong and dangerous allergic reactions. Fungal infections are generally very difficult to treat because, unlike bacteria, fungi are eukaryotes. Antibiotics only target prokaryotic cells, whereas compounds that kill fungi also harm the eukaryotic animal host. Many fungal infections are superficial, that is, they occur on the animal's skin. Termed cutaneous, skin, mycoses, they can have devastating effects. For example, the decline of the world's frog population in recent years is caused, in part, by the chytrid fungus Batrachochytrium dendrobatides. This deadly fungus infects the skin of frogs and presumably interferes with cutaneous gaseous exchange, which is essential for amphibian survival. Similarly, more than 1 million bats in the United States have been killed by white nose syndrome, which appears as a white ring around the mouth of the bat. It is caused by the cold-loving fungus Pseudogymnoscus destructans, which disseminates its deadly spores in caves where bats hibernate. Mycologists are researching the transmission, mechanism, and control of P. destructans to stop its spread. Fungi that cause the superficial mycoses of the epidermis, hair, and nails rarely spread to the underlying tissue, figure 24. 27. These fungi are often misnamed dermatophytes, from the Greek words dermis meaning skin and phyte meaning plant, although they are not plants. Dermatophytes are also called ringworms because of the red ring they cause on skin. They secrete extracellular enzymes that break down keratin, a protein found in hair, skin, and nails, causing conditions such as athlete's foot and jock itch. These conditions are usually treated with over-the-counter topical creams and powders and are easily cleared. More persistent superficial mycoses may require prescription oral medications. Part A is a photo of a red, ring-shaped skin lesion. Part B is a micrograph of long, thread-like mycelia and small, oval sporangia. Part C is a chest X-ray of a person with a fungal infection. Figure 24. 27 fungal diseases of humans. A. Ringworm presents as a red ring on skin. B. Trichophyton violaceum, shown in this bright field light micrograph, causes superficial mycoses on the scalp. C. Histoplasma capsulatum is an ascomycete that infects airways and causes symptoms similar to influenza. Credit a modification of work by Dr. Lucille K. Jorg, CDC. Credit B. Modification of work by Dr. Lucille K. Jorg, CDC. Credit C. Modification of work by M. Rentz, CDC. Scalebar data from Matt Russell. Systemic mycoses spread to internal organs, most commonly entering the body through the respiratory system. For example, coccidioidomycosis, often called valley fever, is commonly found in the southwestern United States, but as far north as Washington, where the fungus resides in the dust. Once inhaled, the spores develop in the lungs and cause symptoms similar to those of tuberculosis. Histoplasmosis is caused by the dimorphic fungus Histoplasma capsulatum. In its human host, histoplasma grows as a yeast, causing pulmonary infections and, in rarer cases, swelling of the membranes of the brain and spinal cord. Treatment of these and many other fungal diseases requires the use of antifungal medications that have serious side effects. Opportunistic mycoses are fungal infections that are either common in all environments or part of the normal biota. They mainly affect individuals who have a compromised immune system. Patients in the late stages of AIDS suffer from opportunistic mycoses that can be life-threatening. The yeast Candida sp. a common member of the natural biota, can grow unchecked and infect the vagina or mouth oral thrush. If the pH of the surrounding environment, the person's immune defenses, or the normal population of bacteria are altered. Mycetismus can occur when poisonous mushrooms are eaten. It causes a number of human fatalities during mushroom picking season. Many edible fruiting bodies of fungi resemble highly poisonous relatives, and amateur mushroom hunters are cautioned to carefully inspect their harvest and avoid eating mushrooms of doubtful origin. The adage, "There are bold mushroom pickers and old mushroom pickers, but are there no old, bold mushroom pickers?" is unfortunately true. Scientific method connection. Dutch elm disease. Question: do trees resistant to Dutch elm disease secrete antifungal compounds? Hypothesis. Construct a hypothesis that addresses this question. Background: Dutch elm disease is a fungal infestation that affects many species of elm ulmus in North America. The fungus infects the vascular system of the tree, which blocks water flow within the plant and mimics drought stress. Accidentally introduced to the United States in the early 1930s, it decimated American elm shade trees across the continent. It is caused by the fungus Ophiostoma ulmi. The elm bark beetle acts as a vector and transmits the disease from tree to tree. Many European and Asiatic elms are less susceptible to the disease than are American elms. Test the hypothesis. A researcher testing this hypothesis might do the following. Inoculate several Petri plates containing a medium that supports the growth of fungi with fragments of Ophiostoma mycelium. Cut, with a metal punch, several discs from the vascular tissue of susceptible varieties of American elms and resistant European and Asiatic elms. Include control Petri plates inoculated with mycelia without plant tissue to verify that the medium and incubation conditions do not interfere with fungal growth. As a positive control, add paper discs impregnated with a known fungicide to Petri plates inoculated with the mycelium. Incubate the plates for a set number of days to allow fungal growth and spreading of the mycelium over the surface of the plate. Record the diameter of the zone of clearing, if any, around the tissue samples and the fungicide control disc. Record your observations in the following table. Results of antifungal testing of vascular tissue from different species of elm. Disc zone of inhibition. Distilled water. Fungicide. Tissue from susceptible elm 1. Tissue from susceptible elm 2. Tissue from resistant elm 1. Tissue from resistant elm 2. Table 24. 1. Analyze the data and report the results. Compare the effect of distilled water to the fungicide. These are negative and positive controls that validate the experimental setup. The fungicide should be surrounded by a clear zone where the fungus growth was inhibited. Is there a difference among different species of elm? Draw a conclusion. Was there antifungal activity as expected from the fungicide? Did the results support the hypothesis? If not, how can this be explained? There are several possible explanations. Learning objectives. By the end of this section, you will be able to do the following. Describe the importance of fungi to the balance of the environment. Summarize the role of fungi in agriculture and food and beverage preparation. Describe the importance of fungi in the chemical and pharmaceutical industries. Discuss the role of fungi as model organisms. Although we often think of fungi as organisms that cause disease and rot food, they are vitally important to human life on many levels. As we have seen, fungi influence the well-being of human populations on a large scale because they are part of the nutrient cycle in ecosystems. They have other ecosystem roles as well. As animal pathogens, fungi help to control the population of damaging pests. These fungi are very specific to the insects they attack and do not infect other animals or plants. Fungi are currently under investigation as potential microbial insecticides, with several already on the market. For example, the fungus Boveria bassiana is being tested as a possible biological control agent for the recent spread of emerald ash borer beetle that feeds on ash trees. It has been released in Michigan, Illinois, Indiana, Ohio, West Virginia, and Maryland. Figure 24. 28. Photo shows a green, stump-shaped ash borer jutting from the bark of a tree with white fuzz, the parasitic fungus, on its body. Figure 24. 28. Fungal insect control. The emerald ash borer, Agrillus planipenes, is an insect that attacks ash trees. It is in turn parasitized by a pathogenic fungus, Boveria bassiana, that holds promise as a biological insecticide. The parasitic fungus appears as white fuzz on the body of the insect. Credit: Hooping Liu, USDA Agricultural Research Service. The mycorrhizal relationship between fungi and plant roots is essential for the productivity of farm land. Without the fungal partner in root systems, 80–90% of trees and grasses would not survive. Mycorrhizal fungal inoculants are available as soil amendments from gardening supply stores and are promoted by supporters of organic agriculture, but there is little evidence as to the effectiveness. We also eat some types of fungi. Mushrooms figure prominently in the human diet. Morels, shiitake mushrooms, chanterelles, and truffles are considered delicacies. Figure 24. 29. The humble meadow mushroom, Agaricus campestris, appears in many dishes. Molds of the genus Penicillium ripen many cheeses. They originate in the natural environment, such as the caves of Roquefort, France, where wheels of sheep milk cheese are stacked in order to capture the molds responsible for the blue veins and pungent taste of the cheese. Part A photo shows a mushroom with a convoluted black cap. Part B shows a pile of sliced mushrooms similar to ones people would see in a food store. Figure 24. 29. Edible fungi. A. The morel mushroom is an ascomycete greatly appreciated for its delicate taste. Credit: Jason Hollinger. B. Basidiocarps of agaricus ready for an omelet, credit. Mary Ann Clark. Fermentation, of grains to produce beer, and of fruits to produce wine, is an ancient art that humans in most cultures have practiced for millennia. Wild yeasts are acquired from the environment and used to ferment sugars into CO2 and ethyl alcohol under anaerobic conditions. It is now possible to purchase isolated strains of wild yeasts from different winemaking regions. Louis Pasteur was instrumental in developing a reliable strain of brewer's yeast, Saccharomyces cerevisiae, for the French brewing industry in the late 1850s. This was one of the first examples of biotechnology patenting. Many secondary metabolites of fungi are of great commercial importance. Antibiotics are naturally produced by fungi to kill or inhibit the growth of bacteria, limiting their competition in the natural environment. Important antibiotics, such as penicillin and the cephalosporins, are isolated from fungi. Valuable drugs isolated from fungi include the immunosuppressant drug cyclosporin, which reduces the risk of rejection after organ transplant, the precursors of steroid hormones, and ergot alkaloids used to stop bleeding. Psilocybin is a compound found in fungi such as Psilocybe semilancida and Gymnopilus junoneus, which have been used for their hallucinogenic properties by various cultures for thousands of years. As simple eukaryotic organisms, fungi are important model research organisms. Many advances in modern genetics were achieved by the use of the red bread mold Neurospora crassa. Additionally, many important genes originally discovered in S. cerevisiae served as a starting point in discovering analogous human genes. As a eukaryotic organism, the yeast cell produces and modifies proteins in a manner similar to human cells, as opposed to the bacterium Escherichia coli, which lacks the internal membrane structures and enzymes to tag proteins for export. This makes yeast a much better organism for use in recombinant DNA technology experiments. Like bacteria, yeasts grow easily in culture, have a short generation time, and are amenable to genetic modification.